Well, hello again from Boston. I'm Arolyn. I'm a postdoc with Tammy Lieberman at MIT, and today I'll be talking about why so many strains of the same species can coexist in a microbiome, in this case, in the human skin microbiome. And broadly speaking, we know that coexistence can arise from adaptive forces or from neutral forces. And adaptive forces include things like niche partitioning or positive selection, whereas neutral forces include things like priority effects and genetic drift. And in addition, we know that spatial structure can also be really important for coexistence. So the main question I'll be asking in today's talk is whether spatial structure arising from host anatomy can shift the balance between adaptation and neutrality. So to explore interspecies coexistence, we chose to study Cutibacterium acnes on human skin for a few reasons. For one, C. acnes is a commensal found on basically everybody at high abundance, and multiple strains of C. acnes typically coexist on individuals. It's particularly abundant on sebaceous skin, which is the oily skin that you find on like your face, chest, and back. And C. acnes is actually found both on the skin surface and inside the follicle of oil-producing skin pores. The skin itself is a great system to study because it's easy to acquire samples over space, which facilitates our study of biogeography. So in particular, we sampled across sebaceous skin sites using different techniques in order to gather material at coarse spatial scale and at fine spatial scale, including from inside the follicles of single skin pores. And for each sample, we streaked out our collected material on an agar plate, incubated it in anaerobic conditions, and picked up to around 15 colonies per sample. We then performed whole genome sequencing of each colony. And the key here is that the sequencing of a single colony gives us information about the whole genome of a single cell in our original sample. And so this sampling process altogether yields multiple full C. acnes genomes from within single follicles. This culture-based approach is critical because it allows us to determine whether pairs of mutations occurred on the same genetic background, which wouldn't be possible with metagenomics. So altogether, this means that we have high spatial resolution down to a single skin pore and high genetic resolution down to a single nucleotide variant, or SNV, across the whole genome. So we want to understand how much strain coexistence exists on our subjects. So after sequencing each colony, we detect SNVs and we group colonies into closely related lineages. In total, we started with around 1,000 colonies from 16 subjects, and these clustered into 53 lineages, which are shown here in this phylogenetic tree. And a lineage is just a set of closely related colonies, less than 100 SNVs apart, found on a single subject. And I've categorized each lineage according to an existing strain typing scheme, so that's what the colors are. And I've highlighted 6 lineages, all 6 lineages from one of our subjects, and I'll be focusing on one subject during my talk today, but you can see data from other subjects in our paper. The fact that we detect multiple lineages means that this individual was colonized multiple times from different sources, and we can make this inference because lineages are separated by hundreds to thousands of SNVs, which is way too much divergence to have accumulated during the individual person's lifetime. And consistent with previous work in the literature, especially from Julie Segre's lab, our subjects tend to harbor multiple coexisting lineages, but individuals don't necessarily have the same strain types present. We want to understand what enables multiple lineages of C. acnes to coexist with each other on the same person. So we'll start by looking at whether they coexist in the same location. Here we're looking at the number of colonies belonging to different strains across facial regions. And the first thing we notice is that strains are not exclusive to specific regions. And maybe this isn't so surprising. Facial sites are close together. Their environments are pretty similar. But you might imagine that there could be niche specificity to the skin surface versus pores, which we know differ in oxygen concentration and nutrient levels. But we don't see a difference between strains we find on the skin surface and inside pores. This is using our 2 different sampling techniques. And so what we've learned is that C. acnes strain coexistence doesn't arise from specificity to anatomical niche. While multiple lineages can be found in close proximity on the skin, they actually don't coexist in the same pore. So to study this, we can look at the probability that 2 colonies originating from the same pore belong to the same lineage. For example, if lots of lineages coexist in the same pore, then when we sample, we tend to get 2 colonies from different lineages. On the other hand, if one colony if only one lineage existed in a pore, when we sample 2 colonies, we'd get 2 colonies from that same lineage. So if the colonies in our data set for subject 1 were randomly distributed into pores, then we'd expect that 2 colonies from the same pore would belong to the same lineage about a little less than half of the time. Instead, what we actually observe is that when we sample 2 colonies from a pore, they're almost always in the same lineage, over 90% of the time. So what we know so far is that lineages appear to be spatially segregated into different pores, even though these pores might be really close together. And what we know is that C. acnes primarily grows inside pores. And so if the lineages are housed in different pores, then they're not competing for the same resources. And this reduced competition between lineages that might have different phenotypes might promote coexistence and provide an explanation for why these lineages don't rapidly outcompete each other. At this point, you should be wondering how we ended up with one lineage inside each pore in the first place. So far, we looked at spatial structure at the interlineage level, and next we're going to zoom in and look at spatial structure at the intralineage level. So right here is an example of a tree of one lineage from subject 1, where each leaf on the tree represents a single colony. And I'm going to go ahead and highlight all the cases where we have multiple colonies from the same pore follicle, and you'll see that a striking pattern emerges. It turns out that not only do colonies originating from the same skin pore belong to the same lineage, but they have very low genetic diversity as compared to the lineage as a whole. And moreover, pores are often monophyletic. So for example, down here we have an example of 4 colonies originating from the same pore that all have completely identical genomes. And up here we have another example of 3 colonies from the same pore, 2 of which have identical genomes, and the third only differs from the others by a single position across the whole genome. And so altogether, this suggests that cells with diverse genetic backgrounds have access to pores all over the face, but that some process is severely limiting within-pore diversity. And we know there's 2 processes that can lead to low diversity. One is population bottlenecking, and the other is selective sweeps. And we don't think selective sweeps are responsible for these patterns. I don't have time to go into the details, but we analyzed the thousands of mutations in our data set that we inferred to have occurred recently on individual people. And we didn't find any evidence of parallel evolution. And we also didn't detect any enrichments of non-synonymous mutations. And these are 2 signals that we'd really expect to be present if selective sweeps are happening regularly inside pores. Instead, we think that pore physiology causes population bottlenecking because it's actually pretty hard for a cell to colonize a pore. For one, a potential colonizer can't proliferate quickly until it gets to the bottom of the pore where the environment is better for growth. Second, it's difficult for a cell to get to the bottom of the pore in the first place. The oils produced at the bottom of the pore flow upward toward the skin surface, and this pushes most of the potential colonizers out of the follicle before they can diffuse into the depths of the pore. And third, once one genotype begins colonizing the pore, the follicle starts becoming crowded, which further slows down diffusion of potential competitors. If you're curious about this, you can check out the mathematical modeling we did in our paper, but basically we concluded that pore physiology in and of itself is sufficient to explain population bottlenecking in pores and why there's such low genetic diversity inside a single follicle. And in fact, the same processes that lead to population bottlenecking also lead to a priority effect, and this has a few implications. The first is that it explains why strains are stable over time, even though the skin is so exposed to the external environment, and that pores protect the extant population. And this resilience to invasion is also potentially relevant for microbiome therapeutic design because it might be important to focus on removing the resident population over optimizing the fitness of your probiotic. So to sum up, we studied C. acnes biogeography on individual people. We found that C. acnes lineages can coexist across individual skin, but not within the same pore. And furthermore, colonies isolated from within the same skin pore are nearly clonal, less than 1 mutation apart on average. And we think neutral bottlenecking rather than selection drives this low within-pore diversity. And this resulting population fragmentation limits competition between C. acnes genotypes. And I think overall, this really highlights how host anatomy can have this profound effect on strain coexistence and competition, in this case, enhancing the role of neutral forces. I want to mention briefly that this work was published this year in Cell Hosted Microbe under their open access option, so please check it out if you'd like to learn more. And finally, I want to thank everybody who contributed to this project, the subjects who participated in our study, and also our funding sources. And thank you to all of you for your attention.